And our final speaker in this session is Ziru Xu, who is currently studying at King's College in London. She's had experience with all kinds of projects, working on traditional Chinese medicine, metabolic syndrome, and clinical medication. However, her interest in lung inflammation and pulmonary delivery of biologic drugs has led her to her current work looking at this interesting topic of the development of inhaled platelet-based therapy for acute lung injury. I'm looking forward to hearing this.
Thank you. Hello, everyone. My name is Ziru Xu. I'm a PhD student from King's College London, supervised by Professor Ben Forbes, Dr. Sam Pitchford, Dr. Magda Swajiska. So today I'm going to talk about development of inhalable platelet-based therapy to repair damaged lungs. So why does platelet therapy attract attention in regeneration medicine? After injury, platelets have a critical role in mediating healing by releasing growth factors from their alpha granulars. They are regarded as a cheap and safe resource of growth factor. To take good advantage of the healing effect, people start to prepare products to concentration the platelet and the platelet-derived growth factors. Related product has already been widely used in plastic surgery, sport medicine, so we are curious whether it is also helpful in lung regeneration. First of all, I want to share a related clinical study. So in this study, they nebulized the platelet-rich plasma directly to the patients with smoke lung injury. The sample they prepared were quite simple. After twice centrifugation of the whole blood, the platelet pellet was suspended in 2 to 4 milliliter plasma and aerosolized this solution directly with the oxygen. But the results were quite impressive. It showed a significant difference of the biological changes in upper airway, halved extubation time, shorter hospital stay, and a less mortality rate. So based on that, we need more lab work to understand the mechanism behind that and try to explore how to nebulize a bioproduct effectively. So my focus is on the epithelium damage because it is a major hallmark of many life-threatening pulmonary disease, such as acute lung injury. And the sample I prepare at this stage is platelet extract. That is to collect the platelet from the whole blood, break the platelet membrane, and dissolve all the growth factors, immunocytokines in the PBS so that they can contact with the cells and the tissues directly. And among different kinds of inhalers, I choose to use mesh nebulizer because it produce less heat and share suggest, and it also requires a small reservoir, small volume in the reservoir, so it is benefit to deliver the bioproducts. So in total, my project starts from the fresh human blood. We collect the platelet, break the platelet, get the platelet extract, and try to nebulize them directly into the damaged lungs. The total aim of this project is to develop a novel inhalable therapy with capacity to restore the lung barrier after injury. I have 3 parts in this project. The first part is about platelet extract preparation. The main question we bring here is how to prepare it easily, effectively, and consistently. And then I tested their effect on the healthy cells and the 2 cell models of lung injury. And in the end, I talk about the delivery by inhalation. I want to prove whether it is feasible to generate a respirable aerosol without losing bioactivity. So the preparation starts from the fresh human blood. After twice centrifugation, the platelet pellet was suspended in PBS. After diluting them into the same platelet level, we sonicated them to break the membrane and dissolve all the things within the platelet in the PBS. Here is my first solution to minimize the variation among individuals, that is to dilute the platelet density into the same level. And after the last centrifugation, I collect 70% of the supernatant as the platelet extract. How could we prove that we prepared this sample successfully? The decreased optical density means most of platelet has already been broken by sonication. Fewer platelets left in the PBS can absorb the light. And most of the growth factors, cytokines, are proteins. So here, we regard all of them as a group. The increased protein level means most of them has already been dissolved in the PBS. Then I tested their effect on the healthy cell line first. The cell line I choose is A549, which is commonly used as an alveolar epithelium type 2 cells. And from the Alamo Blue result, you can see the proliferative effect of the sonicated platelet extract is related to time and concentration. Here is my second solution to minimize the variation among individuals. That is to diluting the protein level into same levels before I applying them on the cells. Then I built 2 cell models of lung injury. The first one is based on inflammation. So our lung is not just a functional organ, but also takes part in many immune reactions. So here, I include an immune cell line that is THP1 monocytose, and LPS is commonly used to induce the inflammation in vivo and in vitro. So I stimulate the immune cells with LPS first, then they can release a lot of inflammation cytokines and fast ligand, and I click all the things in the conditioned medium and treat my cells with the conditioned medium first. From the result, you can see the level of the viable cells were decreased after the treatment of the conditioned medium. And my sample has the ability to reserve the inflammation-induced cell damage. Another model I built is based on wound healing assay. I see the A549 in 24 wells place and leave them for 24 hours to form a monolayer first, and physically create a wound by scratch them with pipette tips, and then treat the wound with our samples. From the photos, you can see the sample of the high protein level can accelerate the wound closure much quicker than the control. The last part is about the delivery by inhalation. The key question here is why the nebulization destroys the bioactivity of the bioproduct. So I tested the samples before and after the nebulization directly on the cells of the same protein level. And you can see there is no significant difference between them. That tells us sonicated platelet extract returns its activity after the meshed nebulization. The last part is about the drug distribution. Of course, we need the next generation impactor. And the mesh nebulizer I use is from Aerogen. And here I use 2 method to recover the drug volume in each stage. The first method is to weight the plate before and after the experiment. That is to focus on the total mass of the droplet. And another method is focused on the active ingredient in the droplet. So I test the total protein again. Both of the methods show us a similar tendency. The mass medium aerodynamic diameter are both around 5 micrometer. Fine particle fraction under 5 micrometer is around 50%. This data shows the aerosol can be successfully to reach the deep lung. So in summary, in the first part, we proved sonication can break the platelet and produce the products easily. And then on the healthy cells and 2 cell models of lung injury, I proved my sample can play a regenerative role on lung cells. And in the end, it is possible to deliver them directly by nebulization. That's all I want to share today. Thank you so much for your listening. I'm happy to answer all the questions.
Thank you, that was fascinating. Have we got any questions from the audience?
Yes, if I may. Thank you very much for your lovely talk. Some years ago, I used to work a lot with the type 1 marker called podoplannin, and I know it's involved in platelet aggregation in other tissues. We also noticed it was downregulated in some forms of acute lung injury, and I'm just thinking whether you have any ideas of the role of this molecule in, I suppose, endogenous platelet aggregation, and whether if you gave the therapeutic form, it may be of interest, or how it would might work. Thank you.
Thank you. So actually, platelet therapy has already been used on skins and muscles tendons, but now we started to try to nucleize them into our body. Personally, I'm very positive about its applications, and today all I share about is healing effect. And we do believe the platelet therapy also can contribute to anti-inflammatory effects in different effects. So maybe in the future we will do more research on different organs and the different types of the platelet products. Thank you.
While I'm waiting for others to move to the microphone, how do you see this being positioned? Would this be something that's frontline in ambulances or in fire engines, or would it be more in the clinical setting?
Oh, actually, because for the traditional development of drug, we start from the lab work and move to in vivo, in vitro, and the clinical. But for the platelet therapy and most of the biotherapy, they start from the clinical. So, yeah.
So we've got one down here.
Hi, thank you. Really interesting talk. It's an area that I know very little about, if anything. So apologies, this is quite a naive question. Based upon what you've done, is there a particular time period where you would need to administer this for it to have the optimum beneficial effect?
You mean the preparation, the duration of the preparation?
Well, following lung injury, if you've got like a sweet spot when you'd need to be delivering this for actually to have an effect, did you do any time course studies of when you've damaged the cells and administer it to sort of understand more about the administration timeline by which this could really maximize its benefit?
From my view, if the patient is really severe, maybe it can be used as a adjuvant therapy with traditional therapy. Or if it is just like the clinical study I share, if it's just a smoke injury, it's not that severe, maybe it can bring some beneficial individually. So I guess it's very independent on the conditions. Thank you.
Thank you very much.
Thank you.
So we are now closing this session of the Pat Bonnell Awards, and I would like to thank all our presenters for their fantastic presentations and their works and for their commitment and efforts on being here. The committee will certainly now have a great task to choose the winner, which we will announce later on, as you know. So I would like to invite you all now to join us downstairs for the break and to please be back at stage at 4:20. Thank you.